We are ready probably to move on to the next talk, a topic that's very near and dear to my heart. We've, I think, as a field, long striven for alternatives to surgery in patients with VHL. A good part of my clinical research efforts are focused on trying to take meaningful systemic therapy strategies into the clinic. Um, as you all know, a variety of studies with VEGF targeted agents have been conducted in this arena. And while effective, there have been serious questions about and concerns about tolerability. So I think we're very fortunate that we now have inhibitors of the HIF2 alpha pathway. The first of these, PD2385, was looked at in a small trial at the NCI. And while there was some evidence of activity, there were some clear problems with this drug, and we quickly moved on to a second generation drug, PD2977, now known as MK6482. And a multi-center study that was presented in part in ASCO and then later at ESMO this year, detailed the results from the study. This was a multi-center effort. The NCI, MD Anderson, MGH, and several other centers around the world participated in designing and conducting the study. So we're very, very excited to hear Dr. Eric Jonasch's update on systemic therapies for VHL. Dr. Jonasch, as you may know, is professor of the Department of Genitourinary Medical Oncology, Division of Cancer Medicine at the University of Texas MD Anderson Cancer Center. Dr. Jonasch.
Thank you very much, and just wanted to compliment you, Ram, for managing this session, and also the entire group for their incredible presentations. It's hard to do these things virtually, but this is really turning into an amazing meeting. Excellent. All right. So here are my disclosures. So, we've heard a lot about what VHL does, and it's a fairly busy molecule in terms of what it regulates. We've heard quite a bit about HIF regulation and the consequences of HIF dysregulation, but VHL also regulates a number of other substrates, and we heard some excellent talks, for example, from Qing Zhang and others, and there's quite a literature sort of looking at how VHL regulates the primary cilium. It likely regulates p53 in a meaningful manner. It regulates the microenvironment by regulating extracellular matrix. And so when we think about fixing broken VHL, the close we can get to the source, and the close we can get to actually remediating or normalizing the VHL molecule itself, the better it would be from a therapeutic perspective. Having said that, though, of course, we have to take what we can get from a therapy perspective, and I'll kind of talk through some of the things that we've done, especially really focusing on the MK6482 study. So, again, this is VHL 101 here. You have a loss of VHL function. You get unbridled heterodimerization of HIF1 alpha, either HIF1 alpha or HIF2 alpha and HIF1 beta, and that results in the transcription of a number of different factors, including VEGF. And so what then happens is you have this tumor cell that is then creating or recreating its microenvironment, and it's going to clearly result in highly angiogenic lesions, but it also influences a number of other things in the microenvironment. And those green cells there are immune cells, and in the sporadic situation and also in the advanced renal cell carcinoma situation, understanding how VHL interacts with the tumor microenvironment is critically important. And this is really a work in progress. And one of the barriers to really doing this in the best way possible have really been the tools that have allowed us to look at human tissue and understand how the tumor cell interacts with the microenvironment. And it's really been through the evolution of things like, for example, bulk or single cell RNA-seq, multispectral imaging, and that we can actually start answering tissue level questions about how VHL mutations affect both the tumor cell in a cell autonomous manner, as well as how it then results in the remodeling of the microenvironment. So how do we block the consequences of VHL loss? So here's a simplified schematic where we have the tumor cells on top and we have the blood vessels on the bottom, and really going to focus just on this sort of simplified model for the purposes of this particular talk. So there has been the development of VEGF inhibitors and VEGF receptor inhibitors as a result of the incredible work that's been done by people like Bill Kalin and others, Othan Iliopoulos, talks you've heard in this meeting, really understanding what the downstream consequences are of VHL loss and the resultant development of multiple therapies that have actually been FDA approved for advanced renal cell carcinoma. But at this point in time in 2020, no therapies have yet been approved for von Hippel-Lindau disease specifically. Moving a little further upstream, obviously, if you can modulate HIF itself, that would be a great thing. And as we've heard from others, these signaling molecules like HIF, transcription factors like HIF, are extremely hard to drug because they're very small and slippery. They don't have easy targets to be able to actually interact with the molecules. And the holy grail really would be to have VHL stabilizers or surrogates. And the word gene therapy is bandied about quite a bit. So I'm going to talk a bit about some of the work that we've done together with others with regards to some of the anti-angiogenic therapies, and then move on to some of the HIF-2 alpha inhibitor. So these agents, VEGF and VEGF receptor inhibitors, started being approved for advanced cancers, including renal cell carcinoma and other cancers in the mid-2000s, really on the basis of the work that's been done and the understanding of VHL biology. What's interesting about these agents is there's both bevacizumab, which is an antibody against VEGF, the VEGF protein itself, but then there's a number of small molecule inhibitors of VEGF receptor inhibitor. And this is a representation just of the kinome. So this is a map really looking at all of the various proteins that are phosphorylated kinases. And what this demonstrates is how very different small molecule inhibitors of various of the kinome can influence this. And although we don't have the relevant protein TKIs for renal cell carcinoma on this slide, just give you the idea of that as a function of these various agents, how they're developed. They result in quite different inhibitory patterns, and these inhibitory patterns are why these agents actually have somewhat different effects on disease biology, and why, for example, with advanced renal cell carcinoma, back in the day when we had sorafenib, sunitinib, and, for example, axitinib, pazopanib available, we could move from one agent to another because they were slightly different in how they affected the tumor microenvironment, and there was utility in trying one agent if the first one didn't work. Because of the availability of these agents, sunitinib was FDA approved for advanced renal cell carcinoma in 2006, and because of the fact that the hypothesis would be that with von Hippel-Lindau disease, where you have angiogenic lesions in the eye, in the brain, in the pancreas, the adrenal gland, and also in the kidney, that blocking the downstream consequences of VHL dysregulation in all of these organ-specific manifestations should be helpful. And so using that, sort of based on that hypothesis, we tried sunitinib, which is a small molecule inhibitor of VEGF receptors 1 through 3, PDGFR, KIT, and other kinases, in a small pilot study of patients with VHL disease. So basically, eligibility for this pilot study were individuals who had germline VHL, proven or clinical evidence of VHL disease, and they received sunitinib 50 milligrams by mouth, 4 weeks on and 2 weeks off, the usual schedule, and they were then RECIST 1.1 was used with some variations actually to measure benefit at 6 months. This was a group of individuals that had a number of different disease manifestations: renal cell carcinomas, hemangioblastomas, pancreatic neuroendocrine tumors, pancreatic cysts, and also retinal hemangioblastomas. And here we see a table that looks at the various organ, how many different lesions were measurable or valuable in the organs. And then what we see here on the third column over is the partial response rate. And what we observe is that there was a 33% objective response rate by RECIST in renal cell carcinomas. But, for example, in hemangioblastomas, although there might have been some very minor decrease in lesion size, we saw no RECIST partial response, meaning a minimum of 30% shrinkage in any one of the target lesions. And the question really is, why did that happen? Furthermore, we also did see some benefit in pancreatic neuroendocrine tumors, and here's a patient who actually was on the trial, had some benefit, had a near PR in one of their pancreatic neuroendocrine tumors, had some central a decrease in enhancement. And then the patient came off study for a while and was then observed to have an increase in size of one of these lesions, and then was restarted in sunitinib. We managed to get sunitinib paid for off study and had re-shrinkage of that particular lesion, and also some renal cell carcinomas, which I don't show here. So one of the concerns that we had with these agents initially was that there would be a rebound phenomenon, that we would treat with these agents, and then the second we took these agents off, there might be an acceleration of growth. Fortunately, we did not see that, and we also did see that re-challenging individuals with these agents might have some utility. Here's an example of a renal lesion that did decrease in size with sunitinib treatment. Unfortunately, and here's a gentleman, and remember this image because this brain is going to feature in a subsequent slide that I'm going to show. This individual, although they had improvement in their renal masses, did not have improvement in their rather extensive hemangioblastomas in their cerebellum. So the question really there, what we asked was, why was that? Why did we see that there was improvement in renal cell carcinomas but not in hemangioblastomas? And you would posit that if that all endothelial cells may be created equal, but it's also possible that perhaps they aren't, and that the endothelial cells in renal cell carcinomas and hemangioblastomas may be different and may have different drivers and different receptors. We do know that hemangioblastomas are very angiogenic. The blood vessels look fairly normal, and the question is, why would they not respond? So what we did in this paper as well was take 20 hemangioblastomas and 20 renal cell carcinomas and really look at the receptor types using a somewhat antediluvian, as of now, technology called laser scanning cytometry to profile the receptors in the endothelia and also the tumors of the hemangioblastomas versus renal cell carcinomas. We saw that VEGF receptor activation state was higher in the kidney tumors compared to hemangioblastomas, but that the phospho-FRS2 which is an intracellular marker for FGF receptor activation, was higher in hemangioblastomas compared to renal cell carcinomas, suggesting that perhaps the endothelia of hemangioblastomas were somewhat more dependent on FGF receptor blockade. Quickly introduce pazopanib, which is another TKI, but one that also blocks FGF receptor. And this is the same individual whom you saw did not respond to sunitinib. And we see, especially looking at panel A versus panel E, this individual, he had some growth in some renal cell carcinomas, and so we managed to get the insurance companies to pay for pazopanib. And lo and behold, what we saw is a dramatic improvement in his hemangioblastomas as well. So no response in sunitinib, nice response in pazopanib, and we reported this just as a case study. But what it did, it is also gave us ammunition to get support for a study to look at pazopanib in a phase 2 study. Once again, individuals who had either germline VHL or clinical VHL that received, it was a non-randomized study that received pazopanib 800 milligrams by mouth per day. And we looked at the primary endpoint was RECIST 1.1 modified with the allowance of 5 millimeter central nervous system lesions at 6 months. This group of individuals was fairly typical for VHL disease. Their median age was in their late 30s. The majority of them had confirmed VHL mutation, but we did allow several that had clinical VHL to come in. The majority of individuals had either renal lesions or central nervous system lesions, and also had pancreatic and retinal lesions. The primary endpoint, as mentioned, was objective response rate, and we saw that in the looking at all of the lesions that were target lesions, the renal lesions, we saw a 49% partial response. We saw a 53% partial response in the 17 pancreatic lesions, and we also saw a modest 4% RECIST response in the central nervous system lesions. Now, it's interesting if you then look at the waterfall plots here. So we have on the top left the renal cell carcinoma waterfall plot, the pancreatic lesion waterfall plot on the right, and the hemangioblastoma waterfall plot on the bottom. And what we see here again, so just as a quick refresher, what these show is the x-axis is a series of essentially bars that represent one patient. The y-axis below the 0 line would be shrinkage, and above the 0 line would be growth. And we can see here with renal cell carcinoma that vast majority of individuals here had some degree of shrinkage in their renal cell carcinoma. Pancreatic lesions, some degree of individuals had some shrinkage in their pancreatic lesions. And then with the CNS lesions, there was about half that had some degree of shrinkage. Now, the challenge with this study or this agent was that it has a number of side effects. It results in the individuals getting a whitening of their hair, which may seem trivial, except with, it's extremely hard to dye your hair. So having, this being sort of a reasonable chronic therapy, that in of itself already is an issue. It results in diarrhea, and this diarrhea can be quite vexing and difficult to manage. It can result in significant fatigue. It can result in nausea. It can result in an elevation in transaminases. And although with sunitinib, after 6 months of treatment with the first study, the large majority of patients said, I'm done with sunitinib. If you had a choice, we offered them a choice between, going off and potentially requiring surgical intervention or staying on sunitinib. They generally chose to get off the study. Here with pazopanib, there was a number who actually decided to stay on afterwards, and we still have patients. This trial was opened in 2011. We still have patients on who are still benefiting. But the majority did end up coming off because this was not something that was really perfect for the patient population. Here are just examples of some shrinkage. We see that the top, we see some renal lesions that decreased in size, some pancreatic serous cystic cystadenomas in the middle. This patient's pancreas was blown up to multiple times the normal size, and then was reduced substantially, and also in the bottom, a hemangioblastoma that decreased in size. So obviously, great, but we need to do better, and so the HIF inhibitor is then what we're currently working on. And just a quick summary here: this agent will block HIF2 alpha, and it was developed at UT Southwestern and then taken over by Peloton, re-derived, and there was a 2385 molecule, and then there was the PT2977, aka MK6042 study. And Dr. Srinivasan presented this at ESMO. It's an update of the data, and this was a 61-patient study. We saw that the primary endpoint was objective response rate in VHL-associated RCC tumors. These individuals had a median age of 41. And amazingly, so we had 92% of patients that were still ongoing treatment with a minimum of 60 weeks follow-up at the time of this reporting, the median follow-up of nearly 70 weeks. We saw an objective response rate of 36% in the renal lesions with an unconfirmed PR of 11.5. So we're getting close to 50% objective response rate in the target lesions here. And you see the waterfall plot on the right. Duration of response was amazing. Very few patients had come off study. And in addition, there was response both in pancreatic lesions as well as in central nervous system hemangioblastomas. So we see that there is a CR of 6.6% and an objective response rate of 64% in pancreatic lesions, 30% in hemangioblastomas, 11% CR, and also in the retinal lesions, we saw an objective response rate in the majority of these retinal lesions. This was well tolerated. There was some anemia, which was manageable, and this agent is now currently under review by the FDA. And we're hoping that it's going to become available in the next 9 months or so. Moving even further upstream, so the holy grail really would be the VHL stabilizers or surrogates, and here there's a fair bit of work that's been done trying to block the things that stabilize or destabilize VHL. There's been sort of talk of doing AAV-based delivery, but this is really in its infancy at this point in time, and much work still needs to be done. So in summary, we have here this axis that we can interact with therapeutically. There are some agents that have been tested blocking VEGF receptors that have provided some benefit, but perhaps not a tolerability profile that is acceptable. We have an extremely promising agent, MK-6482, that's now in clinical trials and hopefully available for patients within the next year or so. Obviously, the work of many, many individuals, the seminal work that's been done by Bill Kael and Othon Iliopoulos from a basic science perspective, colleagues at the NIH like Ram Srinivasan and Marston Linehan, folks at my institution who've helped with the clinical trial side of things, laboratory team, and the Merck team. Thank you.
Thank you, Dr. Jonasch, for that really, really outstanding summary of the state of systemic therapy today in VHL. Looking to see if there are any questions from the audience members. Yes, Dr. King Zhang has a question. He is asking if you think activity that you're seeing with pazopanib, which seems like an improvement over what you saw with sunitinib, might have something to do with FGFR expression and inhibition.
One would assume that it's a possibility. Having said that, there's a study that I did not mention, which was with dovitinib, which was another TKI. We published that last year as well. We had 6 patients on with dovitinib. We saw no response in hemangioblastomas with that, but that could just be it's an even worse drug from that standpoint. So moving forward, I think part of what we need to do is really understand what are the vulnerabilities and what are the endothelial differences between hemangioblastomas and renal cell carcinomas to be able to improve targeting of those particular blood vessels.
Yeah. There's a question about, I think, the potential for gene therapy and whether you'd care to expand a little bit more on whether you think it's feasible. I know you had mentioned it in passing at the beginning of your talk, and I think this question asks for your opinion on where you think this is likely to fit in with other agents that are today available or are likely to be available in the near future for the management of VHL.
So it's sort of an aspirational goal. There's 2 ways that we would be able to do this. One would be through viral delivery, and we know that there already has been a retinal therapy that's been where there's an adeno-associated virus that delivers genes to the eye. And so one could theoretically perhaps engineer an AAV with VHL to sort of normalize hemangioblastomas specifically. Systemic administration, I think, would be challenging. Then there's obviously the so-called CRISPR or Cas9 mediated modification of the genome with regards to whole body use of that. That's really definitely not ready for prime time. So I think we've got some ways to go.
Thank you. I'm going to pose a question to you. You've treated a lot of patients with VHL in your clinic. Question I think I encounter a lot, and I'm sure you do as well, from patients is, you know, what can I reasonably expect in terms of an agent being available for me in the clinic without having to wait for additional clinical trials? I know it's a very difficult question to answer, but I know this is a question in a lot of patients' minds, and I thought I'd pose it to you and get your opinion.
So let me clarify. Are you talking about with regards to currently available TKIs, or are you talking about things that are under investigation?
A therapy that would be widely applicable to the VHL population, and one that we can give to patients off-label or on-label, but without having to find recourse to a clinical trial. A standard of care option, if you will.
Yeah. So I mean, I would say that based on what I've seen with my own patients, I still have some patients who are on pazopanib multiple years later on that trial. I've clearly helped. Some individuals have clearly benefited from that agent. And if you are at risk for losing organ function versus trying that, I think it's reasonable. I frankly also, like for renal lesions, there's nothing magical about pazopanib. The newer generation TKIs like cabozantinib are also things that are worth trying. So I would say that if you have a choice between losing organ function versus trying one of these agents, it's probably worth trying one of these agents.
Thank you. Maybe one last question. There's a question about the role of using HSP90 inhibitors in VHL and the rationale.
Right, and so HSP90 inhibitors would be blocking the agent, a protein that stabilizes VHL, and I sort of alluded to it quickly. I mean, so as we began to understand how HIF was regulated, there was some hope that might actually be a way of destabilizing overstabilized HIF. But the challenge has been that I think the toxicity as well as the off-target effects of blocking such a pleiotropically active agent like or protein like HSP90, it didn't really have the therapeutic index that we were looking for in patients with VHL disease.
Thank you. And if I may add to that, so HSP90 seems to be a good inhibitor of HIF1 alpha, but I don't think there's convincing data that it's a HIF2 alpha inhibitor in the same mold as it inhibits HIF1 alpha. We actually did conduct a study with the HSP90 inhibitor gantenerumab many years ago. It's probably one of the first trials in VHL. Started the study in 2004. As Dr. Jonasch mentions, it was a difficult drug to give for a variety of reasons, but the bigger issue for me was not necessarily tolerability in the population that we studied, at least, but the lack of efficacy. I think the trial was spectacularly ineffective. Only a small number of patients. It was very hard to accrue patients to the study as well. We treated 9 patients, didn't see any responses, and the study didn't go any farther at that point. All right.